Again, cyto refers to cytoplasm, kinesis means division. So, it means division of cytoplasm. Here the first phenomenon for division, each and every cell has to undergo cell cycle. Cycle, cyclic events, a series of events. The series of events, cyclic events by which a cell prepares for cell division. And this particular series of cyclic events by which a cell prepares for cell division, that is known as cell cycle. Now, what are the different steps in a cell cycle? The first phase we can call it as interphase. Second phase is known as M phase. Here you can see the picture of a cell cycle. It consists of again different stages. The interphase, the stage between two cell divisions, where the cell is preparing for cell division. So, it is not a resting stage, it is not a metabolically inactive stage, whereas it is a preparatory stage, and by this stage the cell is preparing for cell division, and it is a metabolically very active stage. Commonly it is known as a resting stage. It is a misnomer. It is not a resting stage, it is metabolically highly active stage, and it is the longer stage in the cell cycle. It will last for about 95% of the cell cycle. So, this particular stage we will call it as interphase. Now, coming to the different stages of interphase. Here you can see the diagram, G1 phase, then S phase, then comes the G2 phase. G1, S and G2 collectively known as the interphase. Then again when you are observing this diagram, you can see that after these three stages, G1, S and G2, there comes yet another phase that we will call it as M phase, with different steps such as prophase, metaphase, anaphase, telophase, followed by cytokinesis. And here there is one more stage represented, that is G0 phase. Some of the cells, even after this preparation, they are entering back to G0 stage, quiescent stage, remains inactive. So, in short, an interphase which consists of three different stages, G1, S and G2. So, these three stages or phases together known as the interphase. Now, coming to the first one, G1 phase. G represents what? Yes, gap phase or growth phase. Gap 1 phase, it is the post mitotic phase. This is M phase or mitotic phase, again G1. So, it is post mitotic phase. Or yes, you see here in the in this cell cycle, here you can see S phase, and before this S phase what you can see? Yes, G1 phase. So, this G1 phase, that is known as pre-synthetic phase. So, synthetic S refers to synthetic phase. So, before that synthetic phase it happens, so it is known as pre means before, pre-synthetic phase. It is also known as growth phase, G1 phase, G4 again growth, 1 first. So, it is known as the first growth phase, and again it is the period between mitosis and DNA replication. During this S phase there occurs DNA replication, and mitosis or M phase, the stage between M phase and DNA replication phase, S phase. What are the events that takes place during this stage? You see here enzymes and proteins. Some of the proteins and enzymes that are synthesized during this stage. Similarly, synthesis of RNA molecules, carbohydrates, lipids, and pooling of amino acids and nucleotides. By using these amino acids, it can make the nucleic acid molecule, that means different types of RNA is synthesized. Then coming to the next point, that is when all these things are synthesized in a cell, naturally the size of the cell will increase. Of course, the size of the nucleus will also increase. A cell in the G1 phase can undergo any of the 3 options. First option, can enter into the next stage. What is the next stage? S phase, and then it can divide. Second option, it may be arrested at any specific point in the G1 phase itself, it can be stopped. The third option is, it may stop cell division and it can enter into G0 stage, and that G0 stage is known as quiescent stage, inactive stage. These cells can again enter back to G1 stage, and it can start just like that of a normal cell depending upon the requirements. So, a cell in G1 phase can adopt any of the 3 options. Now, coming to the S phase, S for synthetic phase, synthesis. What is synthesized here? Yes, DNA is synthesized, DNA replicates, DNA gets doubled. Each chromosome has then 2 chromatids. Then in addition, there needs some of the other basic proteins, for example histone. So, synthesis of that basic proteins, mainly histone, will also takes place during this synthetic stage, S phase. Chromosome number remains the same, even though the chromosome get doubled and each chromosome has 2 chromatids, the number of the chromosome remains the same. Now, coming to the next stage, that is G2 stage, second gap phase or gap second phase, second growth phase also, it is correct, and it is the stage that is coming after the synthetic phase. So, known as post synthetic phase. And this is G2 phase. When you are observing this particular diagram, you can see that it comes before the M phase or mitotic phase. So, we can call it as yes, pre-mitotic phase. Synthesize spindle proteins, the proteins needed for the formation of spindle fibers that are synthesized during this stage. Moreover, there is the synthesis of different types of RNA molecules. Synthesis and storage of ATP molecules, duplication of the centrioles as well as the different organelles needed for cell division, increase in the size of the nucleus. So, by the synthetic activities, the size of the nucleus and size of the cell will increase. Now, coming to the next phase, that is M phase, M for mitosis, mitotic phase or even division phase. It is the stage you can see here comes after these 3 phases. What are the 3 phases? G1, S and G2. You know what term we will give for these 3 phases together? Yes, of course it is interphase. So, it is the stage that is coming after this interphase. So, stage that follows interphase, that is the M phase or mitotic phase. And during this stage M phase, you can see that the nucleus will divide, followed by division of cytoplasm. So, the division of a nucleus we will call it as karyokinesis, and division of a cytoplasm we will call it as cytokinesis. Now, coming to the duration of the cell cycle, whether all the cells are having same duration? No, it varies. It varies from cell to cell, and it also depends upon some of the external factors such as temperature, food and even oxygen supply. Now, here are some of the examples. Bacterial cell, the duration of cell cycle is 20 minutes. When it comes to human cell, the average is 24 hours. Yeast cell, it goes to 90 minutes. And the intestinal epithelial cell, it goes to 8 to 10 hours. So, the cell cycle duration that will vary from cell to cell. Here comes yet another type of cell division, amitosis. It is a simple method of cell division, or it is known as direct cell division. There is no synthesis of spindle fibers. There is no coiling and condensation of chromatin fibers. Nucleus elongates and divides into 2. Nucleus of the particular cell that undergoes amitosis, it will elongate and divides into 2. And after this nuclear division, it is followed by division of cytoplasm. And this type of cell division, it takes place in the primitive type of organisms, mainly for example bacteria, yeast, amoeba, and it can even takes place in the cartilage cells. So, it is the simplest method of cell division or even direct method of cell division. We can call it as direct cell division, followed by elongation of nucleus and then followed by division of cytoplasm. Here is the method. You can see nucleus, it elongates and divides into two by the division of cytoplasm. Now coming to the mitosis. The term mitosis that was coined by W. Fleming, discovered by Strasburger in plant cell and W. Fleming in animal cell. It takes 30 to 3 hours to complete. In human somatic cell, it takes 3 hours. It takes place in somatic cell or even in body cell. So, the cell division that takes place in the somatic cell or in the body cell, that is known as mitosis. It is known as equational division. Why it is known as equational division? Because here after the cell division, we will get 2 daughter cells, and these 2 daughter cells are qualitatively as well as quantitatively remains the same. Moreover, the chromosome number regarding the DNA content, chromosome number and all, it will remains the same as that of the parental cell. So, this particular division, mitosis, it is known as equational division. It helps in growth, repair and regeneration. Here comes to the stages of karyokinesis. Here also the cell will divide by karyokinesis followed by cytokinesis. The different stages of karyokinesis. You know what is karyokinesis? Yes, it is the division of nucleus into 2. The different stages here are prophase, prometaphase, metaphase, anaphase and telophase. So, here there are 5 different stages: prophase, prometaphase, then metaphase, anaphase and telophase. Here you can see some of these diagrams also. Prophase stage, and this is the metaphase stage. In between these 2 stages, prophase and metaphase, there is a short stage, a transition stage, that is called as prometaphase. This is the anaphase stage and this is the telophase stage. Now, we can see what happens in each of these stages, what are the changes, what are the events that takes place in each of these stages of karyokinesis. First one, that is prophase. Here you can see the diagram. Go through this diagram and try to identify the changes, the events, the characters. First one, disappearance of centrosphere. Centrosphere, you know what is centrosphere? The area, the clear area that surrounds the centrioles. So, that centrosphere area disappears. Then what happens to this centriole? What is this particular structure? Centriole. What happens to the centriole? It gets doubled. Centriole pairs doubled and it get moving to opposite poles. It started moving to opposite poles. You can see very clearly in this diagram movement of the centrioles. Now, from each of these centrioles you can see some of the rays fibers they are originating, and we will call these as astral rays around the centriole. The astral rays start developing, and these are the proteinaceous microtubules. Now, what happens to this chromatin reticulum? Chromatin reticulum that undergoes two phenomena. One is coiling. You know, when a structure coils, it gets condensed. So, it undergoes coiling and condensation to form chromosomes. Now, each chromosome, when we are observing, we can see that it has two chromatids. I will show you here the diagram also. Here you can see the chromosomes. Each of these chromosome has two chromatids, and this chromatin reticulum seen in the resting phase or even in the interphase that undergoes coiling followed by condensation, and they forms the thick rod shaped structures. We will call them as chromosomes. Each chromosome there is two chromatids, and here you can see these two chromatids, they are seen attached at the centromere, and during this mid prophase stage, mid phase of the prophase, chromosomes undergo further coiling and condensation. Again the chromosomes undergoes coiling and condensation, that process continues, and finally it becomes a thick and rod shaped structure. Here you can see in the later phase of this prophase, and compare this one with the early prophase stage, there is huge difference. Here these are appearing as even thread like structure, but here it becomes highly condensed thick rod shaped structure. We can call this as chromosomes, and clearly you can see the chromatids, distinct chromatids you can see clearly. So, they becomes more and more thicker, and they becomes shorter, and such thick and rod shaped chromosomes we can see here in the late prophase. Now, what happens to this nuclear membrane? Here you can see an intact nuclear membrane in the early prophase stage, but what happens in this late phase? You see, go through this diagram, the nuclear membrane that starts disappearing. It can be seen represented here as a dotted line only. Here it is an intact line in the form of a definite circle. So, here the nuclear membrane starts to disappear. Now, see these two diagrams, what happens to the nucleolus here and nucleolus here. In the early prophase stage, when you are observing, you can see here the nucleus, clear nucleus you can see, it is visible, but what happens to this in the late prophase? You can see here as dots only, it means it is disintegrating, going to starts to disappear. Now, in a late prophase, there occurs further coiling and condensation of the chromosomes. I have already told you this coiling and condensation of the chromosomes, it is a continuous phenomenon and it goes on, and finally we will get a highly thickened rod shaped structures with the two clear chromatids, we will call them as chromosomes. Now, you can see the asters. What happens to these asters? The centriole with astral rays, they have reached the opposite poles during the late prophase stage. Formation of spindle fibers. Again you see, compare these two diagrams. Here formation of spindle fiber starts. Here it is almost completed. You can see here the spindle fibers that is connecting with these chromosomes. So, formation of a spindle fibers takes place. Asters and spindle fibers, we will collectively call it as mitotic apparatus. You can see here the labeling centriole, the race that is aster, and here you can see the well developed spindle fibers. So, these aster plus spindle fibers, we will collectively call it as the mitotic apparatus. Now, when we are observing, here we can see that there are different types of spindle fibers. One is the chromosomal or discontinuous fibers. The other is continuous or interpolar fibers. Here you can see these different types of spindle fibers. You see some of these spindle fibers, they are attached with this centromere of the chromosomes, and some are moving from one pole to the other, from one centriole to the other without touching these chromosomes. So, such fibers, the spindle fibers that are attaching with the centromere of the chromosome, they are known as chromosomal fibers. Here also you can see the chromosomal fiber, and in between these two there is yet another one, that is the interzonal fiber. It will link the centromere of one chromosome with the centromere of the other chromosome, interzonal fiber. And at the same time the third one, that is continuous fibers, moving from one pole to the other, one centriole to the other continuously without attaching the centromere of these chromosomes. So, continuous fibers extend from one pole to the other. Here you can see continuous fibers, that is moving from one pole to the other. So, they are called as interpolar fibers. Inter means in between. So, in between the two poles, in between the two centrioles, you can see this particular fiber moving. So, it is known as interpolar fibers. Now, here the other one, that is called as discontinuous fibers. They will extend from pole and connect with the centromere or chromosome. They are extending from pole and they will connect with the centromere of the chromosome. This one, they are known as discontinuous. So, since they are connected with the centromere of the chromosome, we can call it as chromosomal fibers. Now, coming to pro-metaphase. Here you can see this diagram clearly, pro-metaphase. A short stage, it is a short stage before metaphase. Pro means before, and it is the stage after prophase. So, the stage between prophase and metaphase, there is a transition stage, a very short stage, that is known as pro-metaphase. During this stage, chromosomes lie irregularly. See the diagram. Some are near the pole, some are in the middle, like that. The chromosomes are seen irregularly on the spindle fibers. Chromosomes are moving towards the equatorial plate of the cell. Equator, that means center of the cell. They started moving, but they have not reached the Some have reached even in the center and some are approaching like that, irregular arrangement. Here you can see the next stage, that is metaphase. Chromosomes are highly condensed one, highly thick one, highly coiled one. You can see it clearly, even you can count it clearly also during this metaphase stage. They are highly condensed and thick one with clearly the chromatids, each chromosome with 2 distinct chromatids attached on the centromere position. So, you can identify it clearly and even you can count it also. Each of these chromosome, as I have already told you, with 2 distinct chromatids. All the chromosomes, see what happens to their arrangement, where you can see exactly at the center of the cell, that is called as equatorial plate of the cell. So, this is the typical character of a metaphase stage, highly condensed, thick chromosomes with distinct chromatids arranged at the equatorial plate of the cell. So, this particular character shows the typical character of a metaphase cell. Now, what happens to the spindle fiber regarding its formation? Spindle fiber formation is completed in this stage. Each chromosome that is connected by 2 chromosomal fibers. This is one chromosomal fiber starting from one pole and this is the other chromosomal fiber that starts from the other pole. Each from each pole and they are connected at the centromere part of the chromosome. Centromere of all the chromosomes, you observe, centromere of all these chromosomes, they lie in the center equatorial plate. Now, the arms of the chromosomes are pointing towards the poles. Yet another identifying feature, centromere will be at the center or even equatorial plate of the cell and arms are pointing towards the poles. Now, coming to anaphase stage. Here you can see the anaphase stage diagram. It is the shortest stage which takes 2 to 3 minutes time. Spindle fiber that will contract. So, contraction of a spindle fiber takes place. Now, what happens when the spindle fiber contracts? The centromere of each of the chromosome will divide. Centromere of each chromosome that will split, that will divide, and each chromatid will move to opposite poles. Here you can see the chromatids and these are the centromeres. So, it will move to opposite pole. The 2 sister chromatids, we will call it a sister chromatid, the 2 chromatids of the same chromosome, that we will call it as sister chromatid. Here also you can see the diagram clearly, or the chromatids, these chromatids are carried to opposite poles, not the entire chromosome as such. So, the chromosome with the 2 chromatid that will divide at the centromere portion and it is moving to opposite poles here. The proteinaceous interzonal fibers, this is what is called as the interzonal fiber, and this interzonal fiber developed between the centromere of daughter chromosomes. Daughter chromosome means the separation